Hi everyone and welcome to our module on drug development. In this module, we'll focus on everything there is to know about the processes involved in the formation of a new drug. And these processes include the process of drug discovery, process of preclinical trial, and then the clinical trial. Drug development is the process of bringing a new pharmaceutical drug into the market. This process typically is regarded as capital intensive and very rigorous. Therefore, it's carried out mostly by large investment pharmaceutical organizations as shown on the screen. So organizations like GSK, like Merck, like Pfizer, like Johnson and Johnson are the organizations that typically would go into the process of creating a drug. Aside organizations like this, government could also be involved in the process because of how capital intensive and how rigorous and how regulatory the entire processes of drug development tends to be. The three major stages of drug development. These stages include the first stage, which is drug discovery, the second being the preclinical trial, and the third stage being the clinical trial. The three stages of drug development tend to span a long amount of years. The average amount of years takes for a new drug to be available in the market tends to be roughly about um, 14 to 15 years. As you can see, the first stage of drug development, drug discovery stage, takes about 4 years. The second stage, which is the preclinical trial stage, takes about 3 years. The clinical trial stage roughly takes about 7 years, is the longest. And then the post clinical trial stage, which sometimes doesn't necessarily occur, is about 1 year. So roughly, the combination of all these different stages of drug development is about 15 years. And this is one of the reasons why it takes a very large organization or the government to be really involved in the process of drug development and not just a single individual. The very first stage of drug development is drug discovery. Drug discovery, as defined, is simply the process by which a potentially new medicine is identified. It involves a multidisciplinary approach, and you see that very large organizations like Merck or GSK tend to employ a lot of professionals, ranging from chemists to biologists to physicists to pharmacologists, and these are the personnel that will work on the drug discovery for them to be able to identify a potentially new substance that could become a drug eventually. And this is just a historical example of drug discovery. On the right hand side of the screen, you could see this is a very popular figure. His name is Alexander Fleming. He was credited to the discovery of the drug penicillin. Now, penicillin during the drug discovery stage um, was discovered to be produced by an organism, a fungi to be specific, a fungal mold, which is shown on the screen over here. So he, as a biologist, discovered that fungi was producing a particular substance, and this substance inhibited bacterial growth. And as a result of various research during the drug discovery stage, the substance produced by the fungi was eventually modified and marketed as penicillin. This is just a typical example of how drug discovery could occur. Now, there are four major stages of drug discovery. These four stages include target identification, target validation, lead identification, and lead optimization. Now, it's not really important that you know this particular stages of drug discovery for your in courses because they're not really high yield. But in terms of explanation, what drug discovery and these four major phases typically entails would be for drugs to be formed. First of all, the substance, what we call the hit or the target, would first of all need to be identified. And once the substance or target has been identified, it's going to be extracted, it's going to be studied. So it's going to go through a lot of screening and chemistry, and they are going to analyze it very well before we can validate its usefulness in a particular treatment we want it to be used in. Then this particular hit or target is now going to be converted to a lead, which is lead identification, and then later on is going to be optimized for better performance, which is lead optimization. Like I said, it's not really hard to know. You probably just see it on a couple of your slides, but they're not really going to test you on it. And this is another picture that historically depicts for us the process of drug discovery. And the drug in this case is aspirin. Aspirin was said to go through the four major stages of drug discovery. The particular target or hit that we identified that eventually was made to be aspirin was gotten from the bark of a willow tree. As you can see on the right side of the screen, you could see a willow tree. What happened was that they scrapped the bark of the willow tree, and the substances secreted from that bark became the hit or the target. It went through different forms of validation and then eventually was converted into a lead. The lead was finally now optimized, and then we got aspirin as a result of the optimization. And this is just how one drug could be going through multiple stages, or could have gone through multiple stages to eventually also eventually arrive at its final form. And as we'll talk about in the next few slides, aside the drug discovery stage, the drug would also need to go through some testings to be sure that it will not be toxic or non-efficacious to humans. Second stage of drug development is the preclinical trial stage. And this is a testing stage, is the laboratory testing stage of new drugs, and is usually done on non-human subjects. The keyword here is non-human subjects. This is done with the hopes that the treatment that we are trying to test really works and it's safe, not just on animal subjects or human subjects, but we would like to test it first on animal subjects before we even test on human subjects. Hence the reason why we're performing a preclinical trial. For the preclinical trial, the experiment is conducted on non-human subjects. This includes both animals alongside human cells. And the goal of a preclinical trial is to evaluate the pharmacodynamics and the pharmacokinetic properties of the drug that we are trying to test. The final stage of drug development is the clinical trial stage, and this is also a testing stage. It's a clinical testing stage for the new drugs, typically done on human subjects. This is contrast to the preclinical stage, which is done on animals or non-human subjects. And the type of experiments that we perform during the clinical trial stage tends to be a controlled experiment. Clinical trial stage involves an experiment that is conducted on human subjects, and like I said before, the experiment would be a controlled experiment. Aside being controlled, the experiment would also be a blinded and randomized experiment. The reason for this is so that we could ensure that ethical rules and regulations are followed, and then the well-being and safety of human subjects being used are also intact. The goal of the clinical trial is to verify three things: to verify the dosage of the drug that will be given eventually, the safety of the drug to humans, and the efficacy of the drug in terms of treating the condition we need it to treat. This is another historical picture of a clinical trial that was conducted some years back, and it was a clinical trial conducted in the United States. It's typically referred to as the Tuskegee syphilis study. In this particular clinical trial, test of the drug penicillin was carried out against syphilis. And what happened was that in the clinical trial, we had some humans being divided into different groups. We had some humans that were blacks, we had some humans that were Caucasians. And what happened, what turned out was that the blacks were not receiving any of the treatment. They were being given placebo and they were lied to. And as a result, most of the blacks that went to African Americans that went to perform the experiment eventually came down with syphilis and eventually had the complications of syphilis and died as a result. And this is just to show that whatever human experiment we'll be carrying out um, needs to be controlled, needs to be following ethical rules and regulations in order to prevent unfairness or injustice being done to humans, in order to maintain ethical principles, in order to maintain the safety of humans. So it was from this experiment that we had a lot or a number of ethical rules and regulations being drafted to just to ensure that whatever clinical trials done on humans next time would be done ethically. Clinical trial involves four major phases from phase 1 to 4. And this is how you to know for your in-course exams. Phase 1 is also called clinical pharmacology phase. Phase 2 is called clinical investigation. We have phase 3 that's also called clinical trial proper, and then phase 4 called the post-marketing surveillance phase. The first phase of the clinical trial is the clinical pharmacology phase of phase 1. In this particular phase, we would recruit or need the human subjects or volunteers to be healthy. Usually, we use a very small number of human subjects, about 20 to 80, or less than 100 human volunteers. And the key word to note about this particular phase is the fact that they are all healthy volunteers, and the goal of the phase one clinical trial stage of phase is to evaluate for the safety of the drug. For the phase two of clinical trial, this is also called clinical investigation. It involves more human subjects, usually 80 to 100. And the difference between the phase one and the phase two would be that in the phase two, the type of volunteers we'll be needing will be volunteers that have the pathology we're trying to treat, that are volunteers that are regarded as being deceased volunteers. So the goal of phase two is to evaluate for efficacy and dosage. As you can see in the picture below, as compared to the picture in the previous slide, if we're to test out a drug that probably would cure toothaches, the set of human subjects we'll be using for phase two will be human subjects that have toothaches, and we'll be using a larger number of human subjects compared to the phase one. Phase three is the clinical trial proper. This involves a larger number of volunteers. These volunteers would also have the pathology we're trying to treat. So they are going to be diseased volunteers. And the goal of phase three is to confirm efficacy and safety. It is confirm the efficacy and safety that we already arrived at in phase one and phase two, but on a larger number of people. Immediately after the phase three is completed and successful, the drug that is being tested is going to be approved by whatever organization is involved in drug regulation. In the case of the United States, that particular organization is the FDA. In the case of Nigeria, the organization is NAFDAC. So once a drug has gone through phase three and phase three has been successful, the drug becomes approved. The final stage or phase of clinical trial is the phase four, and this is also called post-marketing surveillance. This is important to note because it means that after phase three, the drugs are shipped into the market, and the phase four just involves the monitoring of the drug while it is within market, while it's being used by patients with the particular disease we're trying to treat. So in phase four, the subject would be every single patient that is using the drug in the market. And the goal of phase four is to monitor possible rare adverse effects that we did not come across during the first three phases. So this is a historical image that shows the importance of the phase four clinical trial. In the case of this image, the case drug there is thalidomide. Thalidomide previously was prescribed for vomiting, excessive vomiting in pregnant women. It was until later on during long-term research and long-term monitoring that we discovered that thalidomide was teratogenic to the child in a pregnant woman. As you can see, the lower part of the screen, this is a child suffering from limb hypoplasia as a result of exposure to thalidomide during pregnancy. And this was one of the reasons why we eventually incorporated a phase four stage to the clinical trial aspect of drug development to ensure that we are not missing any adverse effects that were not noticed in the first three phases. As a form of summary, the clinical trial stage is the last stage of the drug development, and it's the largest or the longest of the three phases. Clinical trial phase is divided into four major phases, phase one to four. Phase one is also called clinical pharmacology. It involves the use of healthy participants, usually a very small number, 20 to 80. Phase two is also called clinical investigation. It involves the use of a larger number of disease participants, that is, participants with the particular disease we're trying to treat. So about 100 to 300 compared to phase one. Phase three involves a larger number of disease participants, about 1,000 to 3,000. And after phase three, the drugs would be approved or licensed by the particular organization involved in drug monitoring. And then phase four is the post-marketing surveillance phase, and this involves the tracking or monitoring of drugs once they've been shipped into the market to ensure that we identify adverse effects that we did not see in the first three phases. And that concludes our module on drug development.